Welcome to G Fun Facts Online. A quiet revolution is unfolding in medicine, one that takes its cues from nature's own playbook. Imagine microscopic builders, engineered by scientists, that can spontaneously construct sophisticated medical tools right inside your body. This isn't science fiction. It is the burgeoning field of self-assembling nanomedicine, and it's already changing the world. At its heart, self-assembly is the spontaneous organization of individual molecules into stable, well-defined structures without any external director. This process is driven by the fundamental forces of nature that govern how molecules recognize and interact with one another. It's a bottom-up approach, building complex machines from their most basic components, mirroring the way life itself constructs everything from our cell membranes to the double helix of DNA. For decades, medicine has struggled with a central problem: how to get the right drug to the right place at the right time without causing collateral damage. Self-assembling nanomedicine offers a brilliant solution, a platform for creating smart delivery vehicles that can protect their cargo, navigate the human body, and unleash their therapeutic power with unprecedented precision. The magic of this technology lies in its ability to create order from chaos. The process is governed by a few key principles. The primary driving forces are non-covalent interactions. Think of hydrophobic and hydrophilic interactions. Just like oil and water, some parts of molecules are repelled by water while others are attracted to it. In the watery environment of our bodies, this drives molecules to arrange themselves to shield their water-hating parts, often forming spheres with a protected core. Then there are electrostatic interactions, the simple rule of opposites attract. Positively and negatively charged molecules are drawn to each other, guiding them into ordered structures. Hydrogen bonds, the same force that holds our DNA together, and other weaker attractions like van der Waals forces, all contribute to the stability of the final nano-architecture. By mastering these forces, scientists can design nanostructures with precisely controlled size, shape, and function. The COVID-19 pandemic brought the power of self-assembling nanomedicine into the global spotlight. The stunningly fast development of mRNA vaccines from Pfizer and Moderna was made possible by one key innovation: the use of lipid nanoparticles, or LNPs, to deliver the fragile messenger RNA. You've almost certainly experienced this technology firsthand. These tiny lipid bubbles are like microscopic armored cars for the mRNA. The mRNA molecule itself is incredibly fragile. Left on its own, enzymes in our body would shred it in seconds. LNPs form a protective shell, shielding it from destruction and ensuring it reaches our cells intact. But they do more than just protect. Their tiny size and specific surface properties help them get eaten up by our immune cells, the very cells needed to kickstart an immune response. They can also be designed to mimic the size, shape, and surface of a real virus. This tricks the immune system into thinking it's under a major attack, provoking a much stronger and more durable response than a loose antigen ever could. Finally, many of these nanoparticle materials act as adjuvants themselves, substances that boost the immune response, meaning you get the delivery vehicle and the immune booster all in one efficient package. While lipids were the heroes of the pandemic, scientists are exploring a whole range of materials. There are polymeric nanoparticles, made from long-chain molecules, that can be tuned with incredible precision. There are also protein-based nanoparticles, where scientists can design proteins to self-assemble into perfect, virus-like cages, creating a highly effective and repetitive display of antigens for the immune system to lock onto. The success against COVID-19 has opened the floodgates for research into self-assembling vaccines for everything from the flu and HIV to the next pandemic threat. But beyond vaccines, the true revolution may lie in targeted drug delivery. For many powerful medications, especially in cancer chemotherapy, the challenge has always been a lack of specificity. It's a shotgun approach that harms healthy cells along with the diseased ones, leading to devastating side effects. Self-assembling nanoparticles offer a smart bomb alternative, delivering potent drugs directly to the target. They achieve this in two main ways. The first is called passive targeting. It takes advantage of a quirk of solid tumors, known as the enhanced permeability and retention effect, or EPR effect. Simply put, the blood vessels that feed tumors are leaky and disorganized. Nanoparticles of a certain size can slip through these gaps and accumulate in the tumor tissue. Because tumors also have poor drainage, once the nanoparticles are in, they tend to stay there, concentrating the drug right where it's needed most. The second method, active targeting, is even smarter. Here, scientists decorate the nanoparticle surface with targeting molecules. Think of them as molecular keys. These keys are designed to fit specific receptors or locks that are far more common on the surface of cancer cells than on healthy cells. This key and lock mechanism not only increases the concentration of the drug at the disease site, but also encourages the cancer cell to pull the entire nanoparticle inside. But getting the drug to the right place is only half the battle. The other half is releasing it at the right time. Self-assembling nanocarriers can be engineered to be stimuli responsive. This means they hold onto their drug payload until they encounter a specific trigger. These triggers can be internal, found within the disease environment itself. For example, tumors are often slightly more acidic than healthy tissue. Nanoparticles can be designed to be stable in the neutral pH of our blood, but to disassemble and release their drug when they hit that acidic tumor environment. Other nanoparticles are designed to break apart in the presence of specific enzymes that are overproduced by tumors. The triggers can also be external, applied by a doctor for maximum control. Imagine a patient with skin cancer. A doctor could apply a cream containing light-sensitive nanoparticles, and then shine a specific, harmless wavelength of light onto the tumor. This light activates the nanoparticles, causing them to release their drug payload precisely at the site of the cancer, and nowhere else. Similar systems are being developed that respond to focused ultrasound, temperature changes, or even magnetic fields. By combining these targeting and release strategies, self-assembling nanomedicines can make drugs far more effective at lower doses, which means fewer side effects for patients. It's a true paradigm shift in therapy. The versatility of this technology stems from the diverse array of materials that can be used as building blocks. We've talked about lipids, the clinically proven stars of mRNA vaccines, which are great for their biocompatibility. Polymers offer unparalleled tunability, making them ideal for creating sophisticated, stimuli-responsive systems. Peptides and proteins, made from the same amino acids as our own bodies, offer excellent biocompatibility and can even be designed to have their own biological activity. And even DNA itself can be used through a technique called DNA origami to build incredibly intricate and precise nanostructures that can act as scaffolds for other components. Researchers are even creating hybrid nanoparticles, combining a polymer core for high drug loading with a lipid shell for better biocompatibility, getting the best of both worlds. Now, as amazing as this all sounds, the road from the lab to your local clinic is a long one, filled with challenges. A major hurdle is manufacturing. Making a few million perfect nanoparticles in a test tube is one thing. Making trillions of perfectly identical ones for a global supply is a massive engineering challenge. Consistency is everything. Then there is the question of long-term safety. While materials are chosen for their biocompatibility, we need rigorous testing to understand how the body clears these nanoparticles over time. Finally, the novelty of these medicines poses a challenge for regulatory agencies, who are working to develop new frameworks to evaluate these complex products. Despite these challenges, the future is incredibly bright. Self-assembling nanomedicine offers new hope for diseases long considered untreatable. Scientists are designing nanoparticles that can cross the formidable blood-brain barrier to deliver drugs directly to the brain, potentially treating neurodegenerative diseases like Alzheimer's and Parkinson's. The ultimate goal is a future of personalized nanomedicine, where a treatment is tailored to your unique genetic makeup and the specific characteristics of your disease. We are even using artificial intelligence and machine learning to accelerate the design of new self-assembling systems, predicting how molecules will behave, and creating novel materials faster than ever before. The next generation of these particles will be even smarter. Think of nanorobots that navigate the bloodstream, identify diseased cells, and deliver a therapeutic payload, all while monitoring the treatment's success in real time. The journey of self-assembling nanomedicine is just beginning. It's a field built on the elegant principles of nature, aimed at solving our most pressing health challenges. From the triumph of mRNA vaccines to the promise of a future free from once intractable diseases, this is a revolution that is assembling itself, one nanoparticle at a time. Thanks for listening to Fun Facts Online.